All right. Hey guys. Hopefully your anatomy lecture today didn't explode your brain. I heard it was a lot of slides. And your test is obviously coming up this week. So hopefully you guys feel ready for that. And hopefully you do well. I know Justin is trying really hard to help you guys succeed. We all are. So hopefully this first block coming up is good for you guys. So today we're going to cover lecture 7, which is the last part of that human genome PowerPoint. It's mostly the genome browser stuff. And then we're going to cover lecture 8, which is the human genome 2. And we have some questions. And then September 28th for the 5 p.m. is going to be lecture 9, the instability of the human genome. That's your last lecture before, that's counting towards your exam. So that lecture, what's today, Tuesday? That lecture will be tomorrow. And then Monday is going to be just like a Q&A session that he'll hold. So you can come with any questions and he'll answer those questions for you. If you don't have any questions prepared, he's just gonna sit around for a little while and then basically just leave. So if you want to make the most out of that, come with questions for him. And then September 29th will be the morning session for that same last review session. And then September 29th, I will also do office hours from 6 to 8 p.m. for you guys to ask me any questions. And then I will hold last minute office hours the morning of your exam, September 30th from 9 a.m. to 11 a.m. And then again, I'm always available by email. So if you guys don't have any questions before we start into it, then I'll just jump right in. So this morning we had a little bit of an issue with me lagging because one of the PowerPoints I made for you guys is very large, the genome browser PowerPoint. So if that's the case, just let me know and we'll try to figure it out. It was a little stressful for me this morning that it wasn't working, but hopefully we don't run into that same problem today. So let me start screen sharing with you guys. All right. So this is where I left off with you guys last time because I wanted to just keep all of this genome browser stuff together and keep it consistent. So the Human Genome Project is what we did when we identified all of the genes in the human DNA. That's how we figured out the sequence for every gene in the human DNA. So there are 3 billion base pairs that make up human DNA, and that information is stored in databases, which is why we care about Genome Browser. So we have 20,000 protein coding genes. We have 22,000 RNA genes. We have also these pseudogenes and these other kinds of RNAs. So basically what this chart is trying to get at is that there's more than just protein coding genes. So this image kind of makes that point hit home because if you look all the way to the right, that's our protein coding genes for this segment. So it's a very small portion compared to all the extra stuff we have. So we also have our introns. We have our lines and signs, which some of us were a little confused on those last time in my morning session. So they're their own segment on the DNA. They're not necessarily part of these introns. They're lines and signs. And then we have these retroviral-like elements, and we call this transposons because it's DNA moving from using reverse transcriptase. So basically just reiterating that protein coding regions are not the entire DNA, contrast to popular belief. So then here he put the link for Genome Browser, so you guys can click on that. And then he has this list. So after, he already taught this, we're going through it again in this review session. You might have already rewatched that lecture, or you're going to rewatch that lecture. So I would recommend, after you do all that stuff that you want to do, I would recommend going in and trying to do this list yourself. Whether that's after today, or you want to rewatch him again and kind of go through it at the same time as rewatching him, or just go for it after today's session. I would definitely try to go in and do this stuff yourself, because that's how you know, like, you actually understood what he wants you to know. Because, like, I can tell it to you, he can tell it to you, but it helps for you to actually go in and identify what you're looking at. Because I thought I understood it when I went into the first exam. I missed a couple Genome Browser questions, not because they were so hard, but because I was more like passively studying it than actively learning it. So make sure you don't follow my mistakes. It's not too bad once you get into it.
Hey Ivy, for this studying for the Genome Browser, you would suggest like practicing rather than just like hearing, like normally for the lectures I hear like what he says and then I write down exactly what he says, but for this lecture would you suggest more actively practicing than more taking in what he's saying?
So if you watched that lecture, went to that lecture, then that's good. We're going to go through it again during this TA session. So if you like to rewatch it while also going like through it yourself alongside him, I think that would be very beneficial. Not necessarily writing down what he says, because I have written down what he said in your PowerPoint. So it would be beneficial for you to then, maybe if you want to rewatch him, rewatch it and go through what he's doing alongside, like open your Panopto and then open another tab or another window and then just pause and then do it yourself.
Yeah, I got it. Thank you.
You're welcome. Okay, so then he gets into some text regarding what we're going to learn with Genome Browser. So CPG islands are just C's and G's repeated throughout the genome. They're associated with regulatory regions, and they have a role in gene regulation, particularly gene silencing. And it's a methylation on those cytosines that lead to that gene inactivation. So we'll look at that more in detail once we open up the PowerPoint I made you guys. So then here are that list of stuff to do again. And then he's included some text with screenshots. You can look at that after I like the PowerPoint I made for you guys. So then he gets into some of the other browsers that you're gonna use. He more so goes over these browsers like a little more in depth later on in the course. So we're learning about Genome Browser now in exam block 1, but it's not going to go away. He kind of brings it up throughout the rest of the semester. So try to like learn this stuff. I don't remember if it came up on exam 2, but it did come up on exam 3. So although I missed a few questions, I say a few like it was a lot. I missed 1 or 2 Genome Browser questions the first exam, but I was able to relearn it and actually learn it instead of passively learn it for the third exam, and I got those questions right. But he added like a little more detail with these different browsers. So I wouldn't worry about these too much. just what he covered in lecture with you guys, and then just keep in mind that we're going to probably learn more about these throughout the semester if he sticks to what he taught us last year. But for now, just know that COSMIC is a catalog of somatic mutations in cancer, and it's from the UK, which is why I have this random portion highlighted. Then we have cBioPortal. That's for cancer genomics as well. And we don't get the actual sequence. I thought it was listed. Oh yeah, here it is. We don't get the actual DNA sequence because, hello HIPAA. If we got the actual DNA sequence, then we would be able to know like somebody's entire genome, which it's like a violation of privacy. So they only give you the specific mutation. So if it's a specific gene of concern, it will show you the mutant version of that gene. So make sure you know that. And then here I have your fill in the blanks with the answers, so you guys can look at that. Yeah, and then here he kind of talks about what the different web tools are. So I would go through and look at those, especially because I've said it before, I'll say it again, the fill in the blanks do come up on the exam. Sometimes they're not exactly how he presented them to you. So for example, if like this moderately repetitive DNA sequence is like a blank here, it might be like ribosomal RNA genes are blanked out, but it's like this exact sentence. So I would definitely do all of the fill in the blanks before every exam. Our self-study material items, we should not overlook, or would these be in the same category as the yellow highlights he gives us? I would say not necessarily overlook them, but if he didn't care enough to go over them in lecture, it's probably not super high yield. So I would go over points that he talked about like a lot in class first. Then if you feel like you have some time to try to understand those self-study slides, go for it. But they're not super high yield compared to, like, for example, RB, like how we touched on RB kind of in depth. So just like kind of prioritize things a little bit. Because I know you guys have a lot on your plate now. Okay. So you're welcome. So this morning when we were having issues with this, it was lagging when I had it in full screen. So I don't know if you guys want me to try having it in full screen, or if you guys think it will be fine if I leave it like this. Let me know what you guys think.
Hey, Ivy, do you already have this one posted, or is this going to be posted after today?
I believe I already posted it. I'm pretty sure it's already in my modules. If not, I'll do it right after this, but I'm pretty sure it's published. It might help to turn the camera off. Yeah, so I did turn the camera off this morning and it still didn't help. I kind of talk with my hands and explain things with my hands, so I kind of like keeping the camera on. Let's try it and then just let me know if it's still lagging. And then I'll just take it off of full screen. I'll take my camera off and then we'll just go from there. It is posted. Okay, good. So it's published. Okay. So we'll try this. Just let me know. They were saying that my voice was faster than the screen. So just let me know if that happens again and I will adjust accordingly. Okay. So this is the first page that auto-populates when you come to Genome Browser. On the left-hand side, it's going to be the list of species that they have sequenced, and it's like a lot of species that they have sequenced. And it's showing us the conservation from each species. So it's showing us basically how related we are to each species. You know, we evolved from a common ancestor, so however much percent of our DNA is conserved, this region is conserved, this region isn't. This gene hasn't changed in over like 5,000 species, so on and so forth. And then the screen switch, I didn't change anything.
It could just be me, but like I see like the presenting screen down at the bottom and then you at the top. So I'll see if it's my teams, if it's just my teams, but it like switch like from you being at the bottom to you being like in my talking area. But I'll see if it. That's why I was asking if it happened to anybody else because I don't really see it on full screen.
Yeah, it just happened to me.
Oh, okay.
So you're not seeing my screen?
It's just like
Yeah,
very
you're just
small
bigger.
at the bottom, like where normally you would present your screen and I could see your screen at the top and you'd be at the bottom talking.
Yeah.
Like now I see your screen at the bottom. She said try
This-
clicking the left smaller tile. Okay, it worked. Okay, thank you. Yeah,
Alright,
I just have to click
awesome.
on it.
Yeah, we're all just adjusting to this team situation, so no worries. Okay, we got that, but still let me know if I'm lagging. Okay. So at the top, this is make sure you're selecting our newest version. So it says December 2013. He mentioned that December 2013 is not like that new, but that's the newest version that we have. So make sure it's selected to that. And then right under it, position/search term, that's where we can enter the specific gene that we're looking for. So if we're going in order, you can put in item 4 on his list there, which would be chromosome 6, ARMQ, position 21. So you can input that there and search for the gene that you're looking for. So if you put that in there and you click search, this next slide is what will auto-populate when you do that. So if we're looking at this pink arrow that I have at the top, this entire thing up here is going to be your ideogram, which is just the picture or a cartoon of a chromosome. So that entire gray thing is the chromosome. That little red, like hourglass looking thing, that's like the connection of those two arms. So we typed in chromosome 6, RMQ, position 21. So if we look at this little red box over here with this arrow on the right-hand side, on the top right-hand side, that's our Q arm. The smaller one is the P. And in the box, we can see it says 6Q21. So in that box is the position that we are on the chromosome. That's the position that we searched for. And then all of these blue, like under here, those are the different primary transcripts within that location on that chromosome. So all of these blue, like little line looking things with their names next to them, those are all the primary transcripts. So then if we go to the next slide, he tells us to type in a new search term. So he tells us to type in phenylalanine hydroxylase. So we're typing in phenylalanine hydroxylase. He says that's not the best search term because it's not a Hugo symbol, which Hugo symbol stands for Human Genome Organization symbol. A Hugo symbol would be like PAH at chromosome 12. So that's what we give, that's the term we give to the phenylalanine hydroxylase gene. But you don't need to necessarily know the Hugo symbol for the gene you're looking for. You can just type in the name. And if we do that, this next slide is what will auto-populate once you hit enter or search, whatever. So we can see that like we have a bunch of different things here. That's because the genome browser is just like assuming, just compiling everything that we could want to know based off of what we typed in, since we weren't super specific. So it's just guessing like, okay, it's one of these things that you're looking for. So the reason that we have all of these different options is because they're different primary transcripts. So we remember from previous lecture that we define a gene by its primary transcript, so the RNA, and we can include or exclude certain exons and introns. We can have differing start sites, differing end sites. We can have alternative splicing. So there's other things that can cause this variation. So it's the same gene, it's just differing primary transcripts. So then he says, these numbers over here, where I have my pink arrow pointing, are the distances. So if we start at 1, for example, we go all the way to 20. That's just very simplified. If we start at 1 and go all the way to 20, or we start at 1 and go to 10, so on and so forth, he's giving us all of these distances. So he said, you know, if you don't necessarily know what you're looking for, you could just find like the biggest one. So you can have everything all in that primary transcript, and you can do that. But he said, I don't have the patience, so neither do we. We just click on the first one. So after we click on the first one, it auto-populates to this screen. So if we look at the top above our ideogram, it tells us our chromosome 12, and it gives us the positions. So what is that? 1,917,172 to 1,950,850. So, okay, you subtract the larger number by the smaller number and you get 33,000 base pairs. So that's how long this gene goes from the first to the second. If we subtract those, we have 33,000 base pairs that we're looking at. So that's for the entire window, not each specific primary transcript, but for the entire window that we're looking at. So if we want to look for a specific primary transcript, we can see over here on the left-hand side, it says scale chromosome 12, and then we're given those numbers. So wherever you see a start site for your primary transcript, you can look up there and see like a rough estimate of exactly where you are for that primary transcript. And you can see that those numbers continue throughout the screen. So it's letting you know where you are throughout the screen. So I boxed this 102, 917, 172 for like our gene, our start site. So then we can zoom in and we can zoom out. Up here, it says zoom in 1.5 times, 3 times, 10 times, zoom out the same. So he zoomed out times 10. So he just went up here, clicked zoom out times 10. So we zoomed out times 10, and now this is the screen that we are getting. So since we zoomed out times 10, now you can see we have 10 times as many nucleotides that we are looking at. So in this blue box, now we have 336,790 instead of the 33,679. So we have 10 times as many nucleotides on our screen. So is that first nucleotide number is our gene start site for this primary transcript? That is 103 and that is 102, so yes, right? That is 102,920 and that is 102,950. So yes, that would be our start site. See, this is 102,920 and then on the right hand side it's 102,950. Yeah. Okay. So now we have 10 times as many nucleotides. So this yellow arrow that I have on the left, nucleotide numbers for our primary transcript, which is, it's like a little small for me to read that. I don't have my glasses on, but if you guys download this PowerPoint, you can zoom in and see that. And then again, we said that those little blue things are going to be our primary transcripts, right? So if we look, the bottom one is the longest primary transcript, because if you're following my mouse, you can see that we have one end over here, and then we have another end over here, versus all the other ones that are ending right here. So the bottom one is the longest primary transcript. So this is a zoomed in. Why does it look like that? Okay, this is a zoomed in picture of our primary transcript. So on this pink arrow, those little boxes, those are exons. So they're indicated by those boxes. And then the introns are just going to be like the space within this line right here. And these are our untranslated regions. These little top hats are the untranslated regions. I would need to, well, I guess I don't need to. So the arrows are pointing to the left. So that's the direction of transcription. So this is going to be your 3' untranslated region, and we'll get into that in a second for why I know that that's 3'. But that's your untranslated region, this little top hat guy. So then we can see here that we have arrows. I know it's like a little difficult to tell that those are arrows, but they're pointing to the left. So we don't necessarily have a point, but we see that the like wings of the arrows are like back, so it's pointing to the left. Okay, this is kind of blurry, but I don't know why. Okay, so this positive strand and negative strand business was a little difficult for us to understand this morning. Hopefully I do a good job of explaining it to you guys, and you know, you guys can all make sure that your peers understand as well. Because after the business of like lagging and not being able to see and not zooming in, it was just like a little stressful this morning, but hopefully we get this down. So the positive and the negative strand is determined by like genome browser people. So don't worry about why this is positive, why this is negative. How do we know? It's determined by those genome browser people, and we just are going with it. Okay. So in this image, in this PAH gene, we are seeing the scenario on the right. But normally we can see the scenario on the left. So you can see both scenarios here. So I'll start on the left-hand side. If the arrows are pointing towards the right, then the positive strand is the coding strand and the negative strand is the template strand. So if the arrows are pointing to the right, positive is coding. The direction of the arrows are showing us the direction in which transcription is occurring. So that means the arrows are pointing towards the right, and we know we always go 5' to 3'. That means your 5' is on the left-hand side and your 3' is on the right-hand side. And if they're going towards the right, that means our positive is our coding strand. So watch the arrow direction. Yes. So then if the arrows are pointing towards the left, then our minus strand is the coding strand. So if our arrows are pointing towards the left, that's the direction of transcription. So that means our 5' end is on the right-hand side, and the 3' end is on the left-hand side. So what I'm showing you in this image, this is just kind of to try to like understand how like the DNA is orienting itself, because we know we are anti-parallel and complementary. So don't worry about is coding on top, is template on the bottom. Don't worry about that. Worry about the directions that the arrows are traveling in. So we see our primary transcript. So this is not even DNA. It's the RNA that's being transcribed. And it's just showing us the direction in which it occurred. So the arrows are pointing towards the left. So that means that our negative strand is our coding strand. So that means we have 5' on this end and 3' on this end. So stop me if you guys have any questions on that. Okay. If there's no questions, then I'll continue. Feel free to ask questions later on. But yeah.
You said, just to make sure, so 5' on the right side?
That
On the negative, if it's a negative strand, it's on the 5 side. 5' is on the right side, going to 3' to the left.
Yeah, so that's going to be the direction of transcription, right? Because we know we're going from 5' to 3'. So if the arrow is pointing in towards the left, that's the direction in which transcription is proceeding. So we know we have to go from 5 to 3. So that means we started at 5 on the right-hand side, and we're proceeding towards the left-hand side, which is going to be our 3'.
Okay, yeah, okay, got it. Thank you.
You're welcome. Okay. If there is no other questions, then I'm just going to keep going on. Okay. So that would be the template strand. Which would be the template strand, Catherine? The one on the previous slide? No, that's the primary transcript. Remember, we define genes as the primary transcript. The primary transcript is RNA, not DNA, and it's just showing you with the arrows the direction that transcription occurred in. The template strand would be the anti-parallel complementary strand of DNA, if that makes sense. Okay. So in this example, the primary is 3 on the left and 5 on the right, and the template, which the primary came from, is 3 on the right. Yes, Zach, that's correct. So how can the primary transcript and the template be in the same direction in your drawing? Yeah, I just realized I was explaining that. I can redraw this and upload it. I matched the wrong arrow. I labeled them incorrectly. You can't locate the template because they're only showing you the primary transcript. They're not showing you the DNA. They're showing you the RNA that was transcribed from the DNA.
Hey, Ivy, can I state something quickly or ask something quickly?
Yeah.
So is it fair to just say that based off of everything you said, all we need to know is that if the arrows are facing the left, it's negative, and if the arrows are facing the right, it's positive?
Yes. You also need to be able to orient yourself with 5' and 3'.
Okay.
And then he uploaded a document for you guys. So I can go over that after we get through all of the material I have for you. I tried to go over it with the morning people, but I think it was only like 30 people that were still in the session at that time. So maybe spread the word, because I remember getting a test question from that document.
Okay, awesome. Thank you.
You're welcome.
Hey, Ivy.
Yeah.
Really quick, could you go back 2 slides? Just to kind of clarify, the genome browser is only going to show the primary transcript. So every line that we see here is just a different primary transcript, right?
Correct.
Okay, cool.
So it's differing primary transcripts of the same gene because we have different start sites, different end sites, inclusions or exclusions of exons,
Right.
so on.
And so we won't see anything DNA related when looking through a genome browser. It's
You
all
won't,
primary transcripts.
yeah, you won't see like DNA nucleotides. You see the RNA transcribed from those DNA nucleotides, except for when he clicked to those back pages, you can get the sequence, the DNA sequence.
Gotcha. Okay. Thank you.
You're welcome. So, okay. In this slide, the next slide after this, we zoomed out by another factor of 10. So we can still see our PAH gene is there, but now we just have like more stuff on either side. Because we zoomed out more, we can see more genes on either side. So on the next slide, he's explaining the left-hand side of the page to us. So on the top, we have our scale of chromosome 12, because that's what we're looking at. And that has our nucleotide locations, the numbers. And then we see, okay, OMIM alleles, we'll get into those. Then we can see these cons 100 verts. So that means the conservation in 100 vertebrates. So we have conservation of that sequence within 100 different species of vertebrates. We all have the same sequence there. So that is illustrated to you by these little conservation peaks. So they show more conservation, and they usually typically line up with exons. So if we go to the next slide, I boxed it for you. So we see our cons 100 verts. So in this case, it's 100 vertebrates. We have our conservation peak, and we have exons right there in the varying primary transcripts that we have. So yeah, we see a peak lined up with exons. So if he gives you a screenshot on the exam, it might look like this, and then he might block out this cons 100 verts, and then he might ask you like what is lined up in this image, and then you would have to say exons and conservation peaks or something like that. So just make sure that you familiarize yourself enough with the layout of this web page so that like if you look at it long enough, you can memorize it, and when you get an image on the test, you're like, oh, I already know what that is. So go in, do it yourself, review back to this PowerPoint, rewatch his lecture, then you'll have seen it so many times. You're like, okay, I know what this image is. So then now we're seeing the gene names on the left. So that's our PAH gene. Okay. And then now he talks about those CPG islands. He really wants you to know the concept of these CPG islands because he included a slide in the PowerPoint. He went through and toggled this on to explain it to you, and we had talked about it in lecture 1. So I included the slide from lecture 1 where we talk about it. So it's that variant nucleotide where we have a methyl added to this 5th position carbon on the nitrogenous base. This is happening after DNA synthesis, and it's associated with repression. So then this is number 12 on his, or I'm sorry, number 12 is these different designations. This is number 13 on his list. So you would click that, turn it on, you click it to full, and it will show you the clusters of CPG islands. So the C and G is repeated, and then the cytosines are methylated. Okay. So then once you click refresh, after you toggle that on, this is a screenshot of what you would see. Are all the C's in CpG islands methylated? That's a good question. I'm not entirely sure if it's all the C's or if it's one or a couple. That might be a good question to ask him, considering that I don't know the answer. I wouldn't say it's like super high yield to know that on the exam, but that might be a good question to start up a dialogue with him. For the exam, are we just supposed to know the various location of these primary transcripts? Yeah, definitely know the locations of them. So like if you go into Genome Browser and you turn on the settings for the CpG islands, the SNPs, and obviously you'll see primary transcripts. If you turn that on and and turn on OMIM alleles too. If you turn that on and you look at like the full page, you'll basically know where they are. So it might be like one of the top ones, one of the bottom ones. It might be in green, it might be in blue, it might be in black. You'll know that like conservation peaks look like, little mountains, or these CpG islands are just like a little tick mark in green with a number next to it. So I would just think about it like that. Make sure you understand some of the concepts behind it. So like he explained a little bit in detail what CpG islands are. So I would know what that is. So I assume for all these Genome Browser questions, we're given images on the exam. Yeah, he will put screenshots. So you just have to go in and kind of memorize it.
Hi, Ivy. I have a question. Do you think that he would ask, like, what is the number 14th
No.
thing? No. Okay, we don't have to memorize the numbers for those things, right?
No. He just has it like in a list for you so that it's like step by step what you should understand, but you don't have like he doesn't want you to know that list in order. It was just like a list for you to follow.
Yeah, that makes sense. Thanks.
Since CpG and OMIM are both green, they will always be in the same spots to differentiate the two. Yeah, so like when you toggle on the settings, you can see that this OMIM is like higher up. These CpG islands are closer to those conservation peaks. So here in this image, you can see that those OMIM alleles are higher up, closer to the primary transcript. We didn't turn on our CpG for this image, but it'll be closer to those conservation peaks. So, and then you can also like those OMIM alleles, like, okay, here you can see that they're little tiny tick marks. Here, these are bigger with numbers next to them, closer to those conservation peaks. So if he were to show you a screenshot like this, he would like block off the title right here. He would block off the CpGs next to it. If he wanted to show you the OMIM, he would like block off that. Will the images you have on your PowerPoint in last week's quiz be like what he has on the exam? No, it will be like this. I took this. Some images I took from his lecture. Some images I took from when I went in and did it myself. But it's gonna look like this. Okay. So then he talks about SNPs. So SNPs are the single or simple nucleotide polymorphisms, and they're differences in the nucleotide sequences between two individuals within their genomes, but that doesn't necessarily mean there's a medical significance. So it can just be a difference in the allele that states that I have blonde hair, somebody else has brunette hair. Could be that everybody who has type 1 diabetes has a certain SNP at a certain location, and maybe that has something to do with developing type 1 diabetes. So we can use it to try to track disease, but that doesn't mean that every SNP is medically relevant. Although 99% of our DNA is identical. So you could just have some of these variations. So then it's showing you like on the left, I guess, like the number, and it's showing you like the position that the SNP occurs. And then you can see all the way up here, that's our primary transcript. Then we have our OMIM. Then we have, oh, you can kind of see it in this image. Our OMIM is up here, our CPG islands are down there. Then we have our conservation peaks, and then here are our single nucleotide polymorphisms. And all the way at the top is our ideogram. So all SNPs have this RS number. So it's RS with a number following. Don't really know what the RS stands for. I don't even think he knew what the RS stood for. Just know that if you're looking for a SNP, it's going to have this RS number. And let me see if I can, how do I zoom in here? Maybe I can't. But if you look closely at this PowerPoint, you can see that all of these have RS designations. Okay, so here are the OMIM variant alleles. So here you can see like these CPG islands at the bottom right here. These OMIMs are closer to the primary transcript. So OMIM stands for Online Mendelian Inheritance in Man, but he doesn't necessarily like expanding on that acronym because it includes all SNPs, whether they were inherited or not. And those do represent medical problems. So OMIM does have everything to do with genetic disease and so on, whereas SNPs can be, or it can be something as simple as hair color. So that's the title on the right, being indicated by the pink arrow. So if he shows you like a screenshot like this, he would blank out that title and then ask you to maybe identify it. So those are showing you like the different variations within this PAH gene.
Will we just have to know that the OMIMs are just higher up than the CPGs to identify whether it's a CPG or an OMIM in general?
Yeah, you just have to basically like memorize like this image, for example. So yeah, just know that these CPG are located closer to these conservation peaks, thicker bar, number right next to it, slimmer bar, no numbers next to it, closer to the primary transcript. Here you can see primary transcripts on the right. Or if he like expands on them, then it's gonna have like all of this information here.
Yeah, so very detailed. You gotta look at it very detailed. Got it.
Mm-hmm.
Thank you.
If I'm not mistaken, I think like on my exam we had to identify a CPG island, and I know some people are like green, but they're both green, so I wouldn't go about it like that. Just memorize like how that web page looks. I know it seems like annoying, but that's basically all of what your histology class is, so it's good practice. RS equals reference SNP. Okay, nice. Thanks, Kevin. Cool. So then he shows you the homepage of the CBio portal. Okay, awesome. That doesn't really have that much use for us. But then he inputs, wow, what happened? Then he inputs this, we have this RB1 and this BRAF. So the gray boxes are tumor samples from patients. So we're not getting the full sequence. We're not getting that full genome sequence. We're getting just those mutations because HIPAA. Imagine if I could just go on this website and just sequence an entire person's genome. Yikes. So each of these gray boxes are tumor samples, and then so like there's indications below of what each mutation is. So this darker green is like missense mutation, putative driver. Then we have missense mutation, unknown significance. truncating mutation. He didn't really go in depth with, why is it, okay. He didn't really go in depth with making you guys like know the different mutations. I think that might come up later on in the course, but just understand the types of information that you're getting from each database. So this cBioportal is very different information than the human genome browser. So just know the difference there. Okay, and then this is in reference to the next lecture that we're going to go over. These are the back clones. So he talked about that DNA sequencing method and then was so inclined to show you that we can find those on Genome Browser. I don't necessarily think that he will test you on these back clones in the Genome Browser because that wasn't even part of this Genome Browser lecture. I don't even think that that was part of your list for the Genome Browser lecture. I think he just wanted to kind of tie things in together. So I wouldn't necessarily focus on that image, but I just wanted to include it for you because he talked about it. So let me double check. Yeah, that's not even on your list. So don't focus on that. Focus on the other slides. Make sure you go in and do this list and understand it. Okay. Are there any questions before we move on to the questions I have for you?
Ivy, so with the OMIMs being above the CPG islands, is that always going to present like that? Or does it change versus like what's selected? I guess I'm not understanding that part.
Yeah, so they're always going to be in that order if they are toggled on. So if he wants you to identify an OMIM, he might not even put CPG Island as an answer because he knows that that would be like kind of tricky. If not, he would make sure that you could see that it is right below this primary transcript. Or he would show you, let me see, or he would show you like a full image like this, and then he would have the CPG Islands toggled on, and he would just have everything like all of the titles like blocked off. So then you would see, okay, CPG Islands at the bottom near the conservation peaks where OMIMs are at the top. Does that answer your question?
Yeah, so I get that, but for this one that I get the part that you said earlier, but for this one, why would you know that the OMMs are at the top?
Because they are right here on the left, right below this primary transcript. And in this image, the CPG Islands are not, there is no CPG Islands in this image, but in the Genome Browser, they would be above the conservation peaks. So they would be like right where my mouse is.
Gotcha. Okay, thank you.
Yeah. So if he's giving you an image on the test, he would likely either not have it as an option, or he would show you the full image so that you could rule it out. Or he would at least give you a reference point. So if he zooms it into the OMIM, he might zoom it in, but show you that it's directly beneath the primary transcript. So you can associate the OMIMs with the primary transcript and the CPG Islands with the conservation peaks. Those could be your reference points if he zooms in. If not, then you know that OMIMs are above and CPG Islands are below. Okay. General exam question. Are many questions term/definition? Question stem gives definition, term is answer choice. He seems like a, yeah, you could say that. He is a big vocab person. So like, for example, he could say, the unwinding of the DNA for DNA replication is done by what enzyme, and then he would list all of the enzymes, and then you would have to say helicase. So yeah, I would say a lot of his questions are like that. And he was very upfront about it, that he cares about this vocab because he's teaching you genetics and it's like learning a new language. So he wants you to know the vocab, that way you don't get to the test and you're like, I don't understand what these questions are. How many of these browser questions is he giving us on the exam? I'm not sure. Like a few of them. So if there's 9 lectures, there's 50 questions. So then divide 50 by like 9. And then roughly that's how many questions per lecture. Okay. So now we will do this. Yeah, it's pretty evenly distributed. Okay. So as always, feel free to type your answer in the chat so that way you can make sure that we're understanding, and I'll give you a second to type them in before I show you the correct answer. So why are there so many options for the PAH gene? Okay, yeah. So they're different primary transcripts. So different start sites, different stop sites, different exons included or excluded. Introns can be included or excluded. We can have alternative splicing. So all kinds of things going on, which can give us this variation, even though it's the same gene. So what box is being indicated by the boxes in what is being indicated by the boxes in this image? Okay, yeah. Those are going to be the exons. So conservation peaks are usually lined up with what? So that is going to be exons. That was indicated by the pink box I have on your slides. And then CPG islands are the site of what? Yeah, good. That's going to be that cytosine methylation. And we know that that's associated with gene repression. So the gray boxes indicate what? Yeah, so those are going to be the tumor samples from patients. This is the CBio portal. and looks different than Genome Browser, so you just have to kind of know that that's going to be tumor samples. All right. Okay, does anyone have any questions on those before we move on? Does anyone have any Genome Browser questions at all before I move on? All right. If there are no questions, we will just keep chugging along. Okay. So he starts off with definition of an exome, which is going to be all the sequences that represent our exons, versus genome, which is all the sequences we have no matter what. Okay. The first thing we're going to talk about is a DNA sequencing method called dideoxy sequencing or Sanger sequencing. So it has two names, dideoxy because of the type of nucleotide that we're using, and Sanger, named after the scientist who invented it. Because if you invented something this awesome, you're gonna want it to have your name, because scientists are narcissistic. Okay. So yeah, let me know, guys. Is anyone else's screen frozen? Is it me? Can
There is a
somebody
little
else?
bit of lag, unfortunately.
There is. Has it been like that the whole time? Okay, hold on. Let me see if I put this down. We're done with that. Exit that. Save changes. I made a change? No. Okay. All right. Can you guys see it? Am I still lagging? Can you see the screen better now? Okay, you see it now? Okay. Hopefully it was just too many things running in the background and it doesn't keep lagging, because I will rip all my hair out if we have the same issues as the morning people. So on the left-hand side, we are seeing a regular nucleotide. We know we have a 3' hydroxyl, and we know we're always adding new nucleotides to the free 3' hydroxyl group. Think back to like lecture 1 or lecture 2, we're always adding on to our free 3' hydroxyl group. So if we're using this dideoxynucleotide, we don't have that 3' hydroxyl group at all. So if we don't have that 3' hydroxyl group at all, we can't grow our chain, and we terminate the sequence. So no new nucleotides will be added at that point. And we're doing this one template at a time for verification. It has like a high accuracy, so we use it to like verify if somebody has a genetic disorder, but it's not necessarily the most efficient because we're only doing one template at a time. So why is it important to now have this chain terminating nucleotide? So it's going to be covered on this slide, but I forgot he gave you 2 exam questions here, like good examples. So which of the following positions must be missing a hydroxyl group for the dideoxy/Sanger method to work properly? That's going to be the 3' hydroxyl group. So remember these primes. So the first one is going to be our glycosidic bond. That's where we have our base. The 2' is going to have a hydrogen or a hydroxyl group, depending on if it's a ribose or deoxyribose nucleotide. Our 3' always has a hydroxyl group for ribose or deoxyribose, but we don't have our 3' hydroxyl in this dideoxy sequencing method. 4' we don't really care about. Sorry, buddy, you're left out. But 5' has our phosphates. So review this this is important. He loves this topic. He's talked about it a lot. Review this. It probably definitely will show up on your exam. Okay. So then he also gives you another exam question that he could ask you. So he said, I can have an arrow pointing to different parts of this nucleotide, and it could say which part of the nucleotide can be removed without interfering with Watson-Crick base pairing. So he could point to this 3' hydroxyl group, because although we take it out when we use this dideoxyribose ribonucleoside, we can still have our Watson-Crick base pairing with our hydrogens. We just won't add a new nucleotide to the sequence. So now, why is that important? Well, if we have this sequence, this is a sequence that we want, this is a DNA fragment that we want to be sequenced. So, for example, the reason why we might be doing this, we have sequenced the entire human genome, so we know what the sequence should be at a specific location. If we're testing someone to see if they have a genetic disease, then we know, like, okay, this is what should be present, but what is actually present. So if we take a sample from them, we have a DNA fragment and we can sequence it. So since we know what is before that sequence, we can add a DNA primer. So that's what this is in orange. We add a DNA primer. Then we add a normal amount, regular nucleotides. We add like an excessive amount of normal nucleotides. So large amount, regular nucleotides. We add a small amount of these dideoxynucleotides. So if we add a small amount of these dideoxynucleotides and a excess of these normal nucleotides, DNA polymerase will incorporate those dideoxynucleotides randomly. And we can see that, so if we incorporate, so like for example, this piece got unlucky and it incorporated its dideoxynucleotide first. So it was chain terminating at the first nucleotide. So then this one is the second nucleotide, and it was chain terminating at the second nucleotide. So it incorporated a regular nucleotide first and then a chain terminating nucleotide. And then we can see that this last one incorporated it at the third position. Then we can see we're back to a T at the fourth position, and so on. So it will randomly incorporate those dideoxynucleotides at some part of that sequence. So now we can take all of those sequences, we can put them into different wells in this gel electrophoresis. So we know that the smaller nucleotides are faster, they can travel down to the bottom the quickest. So we see the smaller nucleotide down here. The first one we got was an A from up there, so A. Then the next one would be a T, then the next one would be a G, then another T, then a C, then an A. So you can determine the sequence from that individual by interpreting this gel electrophoresis. So here we have A, T, G, T, C, and so on. So those dideoxynucleotides get incorporated randomly at each position. And then we input that into a gel electrophoresis. We see which one's the smallest, which one's the biggest, and then we order them based on size. Then we determine our sequence. And we remember that we have the positive charge at the bottom and the negative charge at the top. And negatively charged DNA likes to travel down to the bottom, which is why you even want to move down this electrophoresis gel. The smaller ones are just able to travel faster through the semi-solid gel. And the chain termination is at different positions because the dideoxynucleotide gets added randomly by DNA polymerase. So we don't necessarily need to do it that way. We don't necessarily need to have them in separate tubes and then put them in separate wells in the gel electrophoresis. What we can do instead is label each nucleotide with its own fluorescence. So if we look up here to the right, we see that A is green, C is blue, G is red, and T is orange. So we can label them with their own fluorescent color. And we can have them all incorporated all together in the same tube and then in the same gel electrophoresis. And then we can generate this fluorescent mapping. And the computer can interpret this mapping. So this peak is the highest here. So that means we must have an A, since A is this green color. And then the computer will determine the sequence. So this is more of a manual version, this first one we talked about, and then the second one is more of using a computer. Would he give us a gel electrophoresis and ask us to interpret the sequence based on it, or is that too much? And instead give a question more so looking to say that we can just, so he could give you both. So for example, he wouldn't ask you to determine this fluorescence mapping. We have computers to do that. It's like a little tricky, but this, like a person can easily interpret that. So he might ask you, it wouldn't be too bad considering that you would just say, okay, this is at the bottom A, this is next T, this is next G. So then you could look for what answer choice starts with ATG. If there are more than one, then you go on to the next nucleotide. So then, okay, A or T, and then C. And then you can just eliminate answer choices that way. But this is like the only method I could see him testing you on, like how to determine the sequence, because this fluorescence mapping, it's too much for the test. He's not going to ask you. And then if we go all the way down, he's not going to ask you to interpret this image either. So if he was going to ask you a question on it, it would be that gel electrophoresis. Okay. You're welcome. So here we have like a compare and contrast of those two different ways to carry out the same sequencing method. So manual over here on the left, we would just interpret it based on the which ones are at the bottom, which ones are at the top, versus this fluorescence mapping. So the peak over here is green, then red, then we have a bunch of green peaks. So the computer will interpret this on the right-hand side. An individual can interpret this on the left-hand side, but you're still getting the same sequence. And you're using different lanes for the gel electrophoresis one, and you're using the same lane for the fluorescence mapping. And the fluorescent labels will not interfere with Watson-Crick base pairing. It's just like a little highlighter to distinguish them from one another. So, did you have a question?
Yeah. For interpreting the gels, will they always, if he were to give us a question on that, would he give us the labels for each, like what nucleotide is in what column?
Yes. So if he were to give you a gel electrophoresis, it would like literally look like this. If you can follow my mouse, it will literally look like this gray square. Then he will have the nucleotide designations, and then he will have like those, like he might just literally copy and paste this image if he is asking you a question like that.
Okay. Thank you.
You're welcome. If we would interpret that, it would be 5'. Yes, so we're doing basically like a PCR. So we have our primer, then we know, based on looking at this, we know A is our first, And that makes sense because if it's our first nucleotide and we get a chain terminating dideoxynucleotide added by DNA polymerase at our first nucleotide, that's going to be the shortest segment of DNA. So the shortest segment of DNA would be our 5' end. The next slide. This is still the shortest, so it would still be our 5'. This is the bottom, so then this A would be our 5'. Okay. So those are awesome. We can sequence DNA like that, but again, keep in mind, I said it's one template at the time for verification purposes. So now we're going to get into like the second generation sequencing technologies. So the example he gives us on this slide is called Illumina sequencing, and they're sequenced in parallel. So we have multiple genomes to be sequenced in parallel. So we can have multiple templates being sequenced at the same time. So second gen and on are going to be massively parallel. So we have our dideoxy Sanger method. We can do it manually or we can do it with the computer. Then we have this second generation, which is Illumina sequencing, which is massively parallel because we can sequence multiple transcripts or, yeah, multiple templates, sorry, at the same time. And then we'll have another example here, which is our next gen, which is almost like a third gen, which is still massively parallel. It's just getting better each time. So we're still able to sequence multiple templates at the same time. So I want you guys to understand that distinction. So basically, we're just getting better each time. So starting from second gen, we have massively parallel sequencing. Okay. With that being said, now let's talk about the Illumina sequencing. I like this picture better. So you guys could read through this to maybe try to like get some more context if you want, but I wrote down what our take-home message is for this. So we're basically doing the same technique as we did in the dideoxy. The difference is we don't have a complete absence of our 3' hydroxyl group. We just have a block. So we can incorporate our 3' blocked nucleotide, which has a fluorescence label on it, into our growing chain. Once we incorporate that nucleotide into our growing chain, we're going to get chain termination. So then we can take a picture and we can you know, have that data of what that fluorescence was. So then we can remove that fluorescence, remove that 3' block. Then now we have our free 3' hydroxyl group and we're able to add a new nucleotide. So then that new nucleotide will have a reintroduced 3' block with a fluorescence label. Then we won't be able to add a new nucleotide after that. It will be chain terminating. Then we take another picture and then we remove that block. We remove that fluorescence. Now we do have a free 3' end. So we can introduce another nucleotide and we just keep doing that and keep taking pictures. And then we compare the different pictures. The computer will analyze that data and put together a sequence. So we can do that with many templates at the same time. So even though we're stopping to take a picture after each individual nucleotide, we're doing that for multiple templates. So it is efficient. So we have a fluorescent attached to our nucleotide. It's not interfering with our Watson-Crick base pairing. We have a 3' block which is different from our dideoxy, but it's the same principle of the nucleotide not being added after that 3' block is added. It's chain terminating. Then we can remove that fluorescence. We can remove that 3' block. So then we do have a free 3' OH. We can add a new nucleotide with its block, take a picture, remove that block, add a new nucleotide, and just keep on sequencing in that manner. So then this is the picture that we are taking. So at each spot, that is where we have the multiple templates that we are analyzing. So like, for example, this yellow one, or like this yellow one right here in the middle that I'm circling with my mouse. At this particular nucleotide in this particular photograph, we have a yellow. So we can then have that data saved in the computer. This is basically a plastic chip that everything is covalently linked to. Then we can wash this off and, not necessarily wash it off, but then we can add our new nucleotides. So wash off our fluorescence, wash off our blocks, add a new nucleotide. It will fluoresce with the color denoted for that nucleotide. We take another image and the computer will track the changes in color at each different location on this little piece of plastic. So we call this PCR in place. Okay. So then now we have our next gen sequencing, which is also massively parallel. It's almost like the third generation that we have developed for sequencing. So chromosomes are a couple 100 million base pairs long, but we can't sequence that all at once. So 120 base pairs is like our limit. So we have to fragment our chromosomes. And we fragment them at different places to generate overlap. We're doing this because we have to be able to put those fragments back together. So we have to say, okay, this is our first fragment, this is our second fragment, it goes at the end of the first. This is our third fragment, it goes at the end of the second. We have to be able to match those back up to understand the data that we're getting for like our chromosome. So fragment them at different places to generate overlap. Then he said, don't worry about this term adapters. We have PCR primers. So we have primers on either end, and they're complementary. So after these fragments are added, they get separated into separate strands. And then each strand will have a known sequence at the end, which is effectively our PCR primer. Then now we can put it on our little plastic chip, this little plastic chip that we had. So we have our DNA, we have our primers. They go like on either end of our DNA sequences. Then we can covalently link our primers to our little chip. So then when we add our DNA fragments, you know, they're binding with our primers. So then we have a primer on one side here, DNA segment, primer on the other side. So if we look at this primer just below the circle in the second image, this bottom primer right here, the purple primer is what's covalently attached to this chip. Then we see that the pink primer is here at the top. So if this just plopped over and bent a little bit, that pink primer at the top can Watson-Crick base pair with this pink primer at the bottom here. That's what we're seeing in this second image. So now those primers are Watson-Crick base paired, right? And then we can add more nucleotides, and we can synthesize using this primer and just use this little arch of DNA as a template. So then now we have a double-stranded DNA molecule because we had our one strand of DNA that like plopped over and those primers Watson-Crick base paired, and then we added more nucleotides and synthesized a new strand. So now we're double-stranded. Then we can denature that double strand. Now we end up with more DNA molecules on this chip. Then we keep on doing that until we get complete amplification. So now we have clusters of all of those DNA segments, and the fluorescents are only added to the sequence in one direction when it comes time to take the images. He said not to worry about this too much, but just understand that it's one sequence that's getting fluorescence. So we're not like mixing them. So then now we can generate this image. So once we've reached like enough amplification here, the camera can like pick up those fluorescents and take that image. And then, you know, we wash it off and do it all over again. So for example, we start with a blood sample. We have 100,000 molecules representing a genome for that blood sample. And again, we know that we can't sequence that many nucleotides at the time, so we have to fragment them. We fragment them at different places so that we can be able to piece those fragments back together later on, because without any overlap, you won't be able to see which fragments go together. Then we do our PCR in place. Then we can do our massively parallel sequencing. The computer collects all of that information, and it will overlap the reads and align the reads to the human reference genome and tell you where the match is not perfect. So the reads are just those 120 nucleotides, like those little fragments. So the alignment has a certain error rate that's like, you know, there to accommodate different SNPs that individuals might have. But if it's past that error rate, then it will tell you, okay, it's not a match. You likely have XYZ genetic disease, like based on the sequence we determined. Okay, that's his source. This one, we didn't really talk that much about it during lecture. It's shotgun sequencing to determine the nucleotide sequence of a whole genome. The genomic DNA is first fragmented into small pieces and a genomic library is constructed, typically using plasmids and bacteria. Okay. So massively parallel sequencing of PCR, he said it's 120, the read length, but it's 120 or 150. So recombinant DNA. So we're using bacteria to sequence that whole genome. Okay. So it's helping us separate out pieces. We can isolate, like the steps are isolate and amplify. So once we have a recombined fragment of, okay. So we have our segment that we want to sequence. We can amplify that in bacteria. So once we have recombined a fragment of a new genome with a piece of bacterial DNA with an origin of replication, that DNA replicates with that origin of replication. So if there's a segment that we want to look at, we can recombine it with bacterial DNA that has an origin of replication, and it will replicate with that origin of replication. So here you can see this red segment, and then now obviously we have many more. And then we can get amplification of that segment of DNA. Then we're able to isolate that DNA from the bacterial DNA. So we're able to take it out basically of that bacterial DNA after, so that we don't have to sequence all the rest of that bacterial DNA that we weren't interested in looking at in the first place. And that bacterium only has one fragment of DNA. So for all of these different colors, we would incorporate them into different cultures of bacteria. So here we have like our vector with our replication origin and our DNA that we want cloned. So we recombine them, they're covalently attached. Then it goes through replication, replication some more, replication some more. Then we're able to isolate that DNA. And after we isolate that DNA, we can do massively parallel sequencing. And then we can piece those fragments together. Again, that's our recombinant DNA. Again. So we call those bacterial artificial chromosomes, back clones. So we sequence lots of different like we have lots of different fragments in those bacterial artificial chromosomes, and we have much bigger overlaps than with reads. So with reads, we said we can only do 120 base pairs. So for example, this one is like 50,000, he said. So that's very reassuring. Like we definitely know that those sequences like line up. Well, that's between 6 and 3, my bad. It should be like between 3 and 2. So much bigger overlaps than with reads. So then when we put those together, we can say like, okay, here's 1 and then here's 2. So this would be our overlap in here where my mouse is. And that could be like roughly 50,000. Okay. And then we like align those overlaps accordingly. So we can know this is our first sequence, second, third, fourth, fifth, and we can continue on until we've aligned all of our fragments that we've sequenced. And then we refer to those as contigs. So he said that definition is not going to come up too often, but to keep it on your list. So this whole thing from A to B, including fragment 1 to 21, that's a contig. So when we're doing that back clone, we're generating these contigs, and we have much more certainty because there's bigger overlap. So this plasmid vector, not too important. This yeast artificial chromosome, not too important, but this bacterial artificial chromosome, this back clone, that's important to know. Okay, and then you can find it on the genome browser, which is why I included that image for you guys in the genome browser PowerPoint. And then here I have the fill in the blanks for you to check your work. Are there any questions on this lecture before we get into the questions I have for you guys?
Hey, Ivy, I had a question.
Shoot.
What is the difference between the bacterial artificial chromosomes and the contigs?
So the contigs are the fragments that we are getting from those bacterial artificial chromosomes that we're making. So for example, this number 1 could be like this red segment And then number 2 could be this yellow segment, and we are amplifying them with our bacteria and then isolating them. And then there's an overlap, and there's a greater amount of overlap than there are with reads. So we're much more certain when we put them back together, and when we put them all back together is when we generate a contig.
Okay, I think I got that. So each of the backs are gonna come from like different segments of the bacteria that we choose, and then the contigs are derived from that.
So let's say we have a DNA sample from somebody that is looking to see if they have a genetic disease. We get that DNA sample from them. We fragment that DNA because we know that we can't sequence it all together at once. With those fragments, we recombine them with a bacterial origin of replication, and we amplify them. So once we amplify them, we isolate those fragments. Now that we've when we recombine them, that's called our back clone, our bacterial artificial chromosome, because where is it? So this recombinant DNA, it's a bacterial artificial chromosome because it's our DNA incorporated into a bacterial chromosome. So it's an artificial bacterial chromosome. But then once we combine all of those fragments that we have amplified and we have sequenced, once we combine them and piece them back together, that is our contig. So we're not putting them back together, like we're not fusing them back together to make another chromosome, but we're piecing them together. So we're saying this fragment comes before this fragment comes before this fragment. And when we're doing that, we generate a contig. Does that make more sense?
Yeah, I think so. So the contigs aren't like, it's just kind of taking each of the back genes and piecing it together rather than a full. Okay, then I got it. Yeah.
Okay, good. Anyone else had any more questions? Okay, there's no more questions. We minimize this so we don't lag again. Okay, and then we'll do these questions, and then we'll go over that document that he has, and then I'll give you guys time to ask me more questions, and then we'll be done. Okay. True or false? There is chain termination at different positions for dideoxy or Sanger sequencing. Come on, guys. Type your answers in the chat. Okay. So that's going to be true. They're at different positions because they get added randomly by DNA polymerase. Those dideoxynucleotides get added randomly by DNA polymerase. So they're not always at the same position. They're at differing positions. So what defines an exome?
Real quick, Ivy, while we're answering this question, would you happen to know why my posts are restricted in TA sessions, but not in like lectures? As in, like, I can't type my answers into the chat.
Oh, really? No, I'm not sure.
And that's only been through like every TA session I've been to, but in lecture I can chat away. And I was just curious if you have heard that from someone before or anything.
No, I'm not sure. It might have something to do with the settings that we. No, because then all students would be restricted. I'm not sure. You might want to like reach out to like Rose, because she was the one who even taught me how to use Teams, and let her know that you're having that issue, because you know it can help you if you just
Right.
want to type out your question or something.
Right. Okay. I can reach out to her. I just thought I'd ask you since you were on the subject of typing in the chat.
Yeah, I don't really know the answer, sorry,
No,
but
that's no problem.
Rose probably will.
Awesome. Thank you.
You're welcome. Are you entering through Teams for TA session? Oh, that's not for me. Okay. So you guys are saying C. Yes, that is the answer. Was this already a question, or maybe I just have deja vu? Okay. In Illumina sequencing, the 3' hydroxyl is blank, where in dideoxy sequencing, the 3' hydroxyl is blank. Okay, yeah. So B, when we're doing our Illumina sequencing, it's just blocked, then we can remove that block and continue to add on nucleotides, whereas in the dideoxy sequencing, it's just missing, so we terminate that sequence and we can't add on nucleotides after it's been incorporated. So true or false, each bacterium has more than one DNA fragment when generating bacterial artificial chromosomes. So that's going to be false. Each bacteria just gets one fragment. We have different like agar plates that we incorporate one fragment into one bacterial colony, one fragment to another bacterial colony. They get amplified. We then isolate the amplified fragments. We do our massively parallel sequencing. Then we compare those sequences. We form our contig when we piece them together at the end. Which sequencing technique that we learned about has the largest overlap between DNA fragments? Yeah, so that's going to be those back clones, which is why we like using that method. Okay, that is all for my practice questions for you guys. Feel free to ask me any questions, and we can also go over this document that Dr. Blank has. So I'll give you guys a second to ask me any questions before I go over this. If there's no questions, then I'll just jump into it. Okay, looks like there's no questions. So this document is kind of important to understand. So I looked at it like super last minute before my exam, and it happened to be a question on the exam. So kind of glad that I did that. But okay, let's go through. So this image represents the 3' end of the PAH gene as shown in the display of the genome browser. This image is highly focused at the 3' end, and at this level of resolution, the genome browser displays what is referred to as the positive strand. So the genome browser is showing us the positive strand, which is why we have the designation of the arrows in different directions. So if the positive strand is the coding strand, arrows are pointing to the right. The positive strand is not the coding strand, arrows are pointing to the left. Okay. So now he's telling us, note this particular nucleotide sequence in that red oval. So, okay, we see that we have T, 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 so 3 T's, CTG, 5 T's. And then he's telling us the direction of transcription because for whatever reason in this screenshot, we don't see the arrows on our primary transcript. It's just a block. Might have something to do with the resolution that we're at. But he tells you the direction of transcription. So to the left. So if it's to the left, that means our minus strand is our coding strand. So we know if this is the positive strand, this is our template strand. Well, this is our primary transcript, but showing the primary transcript of the positive strand. Okay. So there are 2 sequences in the pages below. The first sequence represents the PAH gene's mRNA. Note at the end of the mRNA within the yellow highlight. Compare the sequence in the yellow highlight with the sequence in the red oval on the preceding page. You will see that the sequence on the preceding page is the reverse complement. If I want you to remember one thing about this document, it is reverse complement. I remembered that. That's what got me the correct answer on the test. Reverse complement. So the sequence on the preceding page is the reverse complement of the yellow highlighted sequence, meaning that the sequence in the red oval is used as the template. So now let's scroll down to our sequence. Okay. So above we had said 3 T's first, and then what did I say? 3 T's, CTG, 5 T's. So now if we look down here, we have 5 A's, CAG, 3 A's. So it's the reverse complement of this sequence because we know that this is our template. So if this is our sequence, we would have the, this is the direction of transcription, we would have the reverse complement. So 1, 2, 3, 4, 5. So we're going in the opposite way. So this is our direction of transcription. So we'll have A with a T, 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 C with a G, A with a T, G with a C, A. Do we see that? A, C, A, G, A. Yes. So just remember the reverse complement. So the following sequence is the gene sequence for the PAH gene, available from the back page. The sequence is presented as the negative strand for the reference genome. So see the top of the next page, it says negative in the yellow highlight. So just as in the case of the mRNA, the end of this sequence is the reverse complement of the sequence displayed in the first page of this in the red oval. So he gave us all of the lovely sequence, like 30 pages of this sequence. We don't care, we don't care, we don't care. Still don't care about all this. But what we do care about is this. So we still see that it's the reverse complement. We have 5 A's and then CAG with 3 A's. That's because it gave us the positive strand. It gave us the primary transcript for the positive strand, but our arrows are going in the left direction, which means that our coding is actually the minus strand. So you have to take the reverse complement of it. That's all you need to know. If I remember correctly, it gave us like literally this sequence literally this sequence in red right here, he gave it to us on the test. And then he asked us basically what is, basically, I don't remember what the question said, but the answer was this yellow highlight. So he could have given us like this picture with the arrows and then say, what is the coding sequence? And what is the coding sequence? And if you know, the arrows are pointing to the left, this is denoted as positive, but the arrows are pointing to the left, we know our coding is negative, so we have to take the reverse complement. Okay, did that make sense? Do we have questions on that?
Sorry for all the questions, but
No worries.
how in this case would we identify the which ones are positive and negative in this picture?
So they're always going to give you the primary transcript for the positive strand. But if he was going to ask you a question like this, he would denote that this is the positive.
Which one were you pointing at for the positive?
So these are primary transcripts. So these are not DNA sequences. So this is not a positive or negative strand. This is the primary transcript from the positive DNA strand. The genome browser is always going to give us the primary transcript from the positive strand, which is why we have those difference in arrow directions. So if we're pointing towards the right, that means that positive strand that they're giving us is the coding strand. But if the arrows are pointing to the left, that means that positive strand that they're giving us is the template. And they're giving us the primary transcript, not the actual gene, like nucleotide sequence.
I got it. Okay, yeah, that's on me. Thank you.
No worries. That's a little confusing. I know I struggled with this, so are there any more questions?
Oh, yeah, I was just gonna say, I think for this one though, I think why he's a little bit confused is because we're given the two primary transcripts, but we don't see the arrows, right?
Yeah. So that's what I was saying earlier. Like this blue box, like this primary transcript doesn't show the arrows, probably because of the magnification that we're in or something, but that's why he gave you this blue arrow at the bottom and said the direction of transcription. So if you get a screenshot like this on your test, he will make it so that the arrows are showing, or he will make it known which way the direction of transcription is going. He will make it known that this is the positive strand, what's the negative strand, something like that.
Okay. So if we're just giving the negative strand, just switch up the original sequence that's given and call it a day, right? Like
yeah. So my question on the test was something like, he gave us this sequence in the red circle. Then he like probably denoted this as the positive strand, but the arrows were pointing to the left. So then if I know the arrows are pointing to the left, the positive is not the coding, the positive is the template. So I have to take the reverse complement of that.
Okay, got it. Thank you.
Okay, good. Does the genome browser always display the positive strand? Yeah, I'm like 99% certain that the genome browser always gives us the positive strand transcript, because that's the whole point of having the different direction of the arrows. Yeah, and then he has it here. The genome browser displays what is referred to as positive strand for the reference genome. So the coding strand will be flip it. So you did the complement, but you didn't do the reverse complement, Sarah. So the direction of transcription goes in the other way, Sarah. So if we scroll down here, we have our 5 A's first and then our 3 A's last.
Is the hi Ivy, is the yellow 5 to 3 direction? So the 5 A's in the beginning, what you highlighted, is that 5 to 3?
Correct.
Okay. And that's basically just the reverse of the template that's above.
Yep, because we know that we are anti-parallel.
Okay. Perfect. All right, thank you. It's a little confusing at first, but now thank you for clarifying everything.
Yeah, of course. I know when I was in your shoes, I struggled with this like for a hot minute, which is why I just barely looked at this like right before the exam, but I'm glad I did because it was literally an answer.
I just have one more quick question. For the direction of transcription, how it's pointing to the left, that's how we're supposed to know our 5' and 3'. So
Correct.
So that's basically saying that our 3' is on our left.
That is saying our 3' is on our left.
And that's why when we take the reverse of that, the 5 is on our, like,
right.
Okay. I know I'm confusing myself just talking about it, but I think I get it in my brain.
Okay.
But,
So just remember, like,
yeah.
if you get that on the test, just do the reverse complement.
Okay, reverse complement. So take that, reverse it, and then just put it 5 to 3. Okay.
Mm-hmm.
Thank you so much.
You're welcome. You're welcome. Okay. Any other questions, guys?
I have a question. So since it's the reverse complement, is that why it's 5 to 3? If it wasn't the reverse, then it would have been 3 to 5?
If it's the reverse complement, why Sarah's answer? Is that what you're asking?
Just about the 5' to 3. So like in what instance or like what type of question would the correct answer be 3 to 5, if that makes sense?
We always are going from 5' to 3'. There's never, ever, ever a case where we're going from 3' to 5'.
Okay. Thank you.
You're welcome. What I was mentioning was that we have like, there's 5 A's here. That's what I was mentioning. And then our 3 A's at the end. But there's always 5' to 3'.
Right. Okay. I understand now.
Okay. Any other questions, guys?
I have one for you, Ivy, really quick. I understand the whole 5' to 3' reverse transcriptase, all of that. Do we need to make sure we differentiate between U's and T's because the primary transcript is RNA, right?
Yeah.
And
it is. Yeah. It is RNA, but the transcript that he gives you does have T's.
Okay. So do you think we would need to look out for that in the answers? Like, would he put like T's instead of U's if necessary?
No, he's not going to trick you like that. He really is like a fair tester. And when I'm telling you that like this was my exact answer, this highlight was my exact answer. I can't guarantee that he's going to use the same question for you guys, but I'm just throwing it out there that this was my exact answer.
All right, I understand. You're just giving a little hint here and there. I understand.
Yeah, but I can't guarantee. Like I don't write his tests. I know they have like a test bank, so they can pull different questions from year to year, but that was on my test.
Ivy, is there a reason why some of them are lowercase and some are uppercase?
I'm sure there is, and I think it was that the uppercase was the introns and the lowercase was the exons. Yeah, because they're lowercase in here, and then we get into uppercase, and then, yeah, I think that's the reason. Don't stress on that.
Okay, sounds good. Thank you.
You're welcome.
Can I add to that real quick?
Mm-hmm.
I believe, don't quote me on it, I'm pretty sure he said the uppercase were the exons and the lowercase were the introns, because the exons were more important to us, so they were uppercased. I could be wrong, but I vaguely remember him saying that. Because I remember him looking for uppercased ones when he was going through the transcript in class, and he couldn't find any.
Yeah.
Unless this one's backwards, but...
This one might be different because what we're looking at of importance is lowercase. There was a toggle between which ones you want. Okay, so I guess we can switch it up. Yeah.
Awesome.
So a lot of the people this morning left before I went over this. So maybe like spread the word, tell them to look at this, help them out if they have any questions, because, you know, if you can teach it to them, then that means you like got it down. I like had, what did I start with? 113 students. I think I was down to like maybe like 40 students or something when I went over this. So that makes me kind of sad, and they might miss an exam question, but maybe let them know. And other than that, I don't have anything else for you guys. I can stick around for like last minute questions, but yeah, that's all I have for you guys. So good luck on Thursday, and I'll see you guys on Friday.
Thank you, Ivy.
You're welcome. You're welcome, guys. You're welcome, guys. Okay, so I guess there's no other questions, so I will see you guys on Friday.